Good afternoon and welcome to the last PET lecture of 2020. It's been one hell of a semester. Well done for getting through it. Very proud of you all because it's been tough. So we're finished with something that's a little bit cheeky to understand, and that's muscle physiology. So although it won't actually we won't require the huge amount of detail that we go into today. It's really important that we understand the background of muscle contraction when we're prescribing exercise. So I just want to make sure everyone can hear me in the chat. Anyone that's just got into the room, just give me a thumbs up in the chat if you can see and hear me, please. Just before I proceed, make sure I'm not talking to myself. Quick thumbs up in the chat if you can see and hear me, guys. 2 of you, 3 of you. That's better. Okay, I need one more. That will do, that will do. Okay, fantastic. Good, just to make sure that I'm not speaking to myself. So what are we going to cover today? So we're going to cover lots of things. This is our centering slide. Obviously, we're going to be focusing on human anatomy and physiology, human movement and function, maximizing wellness and potential, minimizing disease and injury, and solving real-world problems. That is what our profession is about. We come back to that statement time and again so that we know exactly what we're doing. We've got a sport Billy that we know tirelessly. Okay, we don't need to go through any of that. You're all good with that. That's fine. So what we're going to cover today, we're going to cover the properties and the organization of muscle tissue. We're going to describe the physiology of muscle contraction and explain the sliding filament theory. Now, normally we do this through sort of physical theatre. So there's a little video for you to watch at the end in between. So we're going to be doing 2 sessions today, muscle physiology, and then the revision session as well. So 2 hours jam-packed full of PET goodness for you. Lots to look forward to. Okay, so there's a little bit of directed study that I snuck in. It's really for you to just understand more about muscle contraction. You're not just going to understand muscle contraction in an hour. It's going to take a bit of time for you to get your head around these concepts as well. So I've got a series of videos for you to watch on muscle contraction that will hopefully just make it much, much easier to understand. And then what we're going to do is we're going to go through this sort of quick overview and understand what muscle physiology is all about. How is a muscle contraction produced? And just have a greater understanding of what we mean when we talk about muscle contraction, which is ultimately quite a lot of what we do in physiotherapy. So we're going to explore the events that lead to a muscle contraction. We're going to explore things like length, tension ratio of the muscle, and of course the sliding filament theory. Just give me a thumbs up in the chat if you've already done the sliding filament theory, either on a college course, A-level, B-Tech, or whatever. Yeah. Okay, so this is going to be very similar to what you've done already, so there's nothing to worry about here. Okay, so let's talk about muscle tissue. So muscle tissue is remarkable because ultimately with a stimulus, i.e. with a training overload stimulus, we can get muscle tissue to grow. And we can get muscle tissue to grow almost at any age, which is unbelievable, which means you can strength train up until the day that you pass. Okay? Now, the reason being is because muscle tissue has 3 properties. The properties of excitability, conductivity, and contractility. So what do we mean with all these 3 tissues? It's basically allowing this sort of electrical energy to be converted into chemical energy, which will be converted into mechanical energy. Easy for me to say. Okay, so we've got electrical stimulus, okay, happening down the action potential in the nerves. That will then change into the chemical energy as various neurotransmitters get released, and that will then lead to mechanical energy in the form of muscle contraction. So when we talk about the muscle tissue, so it's excitable, which means that there is chemicals that can be released from nerve cells. And conductivity is basically the ability of this muscle to propagate these electrical signals And then we've got contractility. Basically, muscles shorten and muscles pull. Okay, so they never push, muscles always pull. What is the only muscle in the body that pushes? All other muscles pull. What is the muscle in the body that pushes? Just type it into the chat, see if you can guess. What's the only muscle in the body that pushes? Not the heart. The heart will still squeeze. Okay, but I like the fact that you're thinking like that. What's the only muscle in the body that pushes? Not the diaphragm. I like these. I'll give you a clue. It's in your head. Somewhere in... It's the tongue. Yes, Will. It is. Yes, James. Okay, so put the tongue in the roof of your mouth and push it against there. So the tongue is the only muscle that pushes. Okay, well done. That was good. Some of you heard that before, I'm sure. Okay, so we look into the muscle fiber. So if we deep dive into the muscle, imagine look at it through the microscope, we're going to find muscle cells. They're long, cylindrical, and then they've got multi-nucleuses within them. So we refer to the muscle tissue as the sarcolemma, okay, or the sarcomere, and that's the official name for muscle tissue. And it's filled with sort of tiny threads, myofibrils, and myoglobin. So that's what a muscle looks like under the microscope, with a bit of detail there. Okay, so what we want to understand is we want to understand the sliding filament theory. So over here, we've got what muscle looks like under the electron microscope. And then we've got the diagram of kind of what's going on here. So here we've got two muscle tissues, two sarcomeres here. And what we need to know is that a sarcomere is basically Z disc to Z disc. That represents, okay, a sarcomere or one sort of motor unit of muscle. Within that, down the center is the M line. Yeah, so we've got Z disc to Z disc. And then we've got the M line denoting the middle of that. Here we've got the H zone. Here we've got the A band. And then we've got the I bands. Don't worry too much about that. Just orientated you to what they are. Okay, so is understanding, get a little bit of muscle physiology here. So this is what the muscle looks like when it's relaxed. Okay, this is what the muscle looks like when it's partially contracted, and this is what the muscle looks like when it's fully contracted. Okay, so that just gives you an example of what's going on. And we've got the fibers sliding over each other. So they slide over each other. That's how they work. Okay, they don't sort of concertina up like we used to think. They slide over each other. And that's the sliding filament theory between the actin and the myosin filaments. Okay, so basically proteins within the muscle, and there are three kinds of protein that we've got. So we've got the contractile proteins, and these are the myosin and the actin. Then we've got the regulatory proteins, which basically switch the muscle on and off, and that's troponin and tropomyosin. Then we've got the structural proteins that basically provide alignment, elasticity, and extensibility of the muscle. So we've got titin, myomycin, nebulin, and dystrophin. Okay, so you might have heard of the term muscular dystrophy, and that's with a certain lack of this structural protein. So it's just something to bear in mind there. So here we've got the myosin and the actin filaments here. So we've got the thick filaments. These are the thick filaments, and we've got the thin filaments here. Okay, and then we've got the troponin and the tropomyosin here, and then we've got an entire sarcomere, and then we've got the sliding filaments. Okay, more as a kind of pictorial of what happens. Okay, so it's easy to get lost here, so don't worry too much. Okay, but are we happy so far? Have I lost anyone with regard to thick and thin filaments, troponin and tropomyosin? Just give me a thumbs up in the chat if you're still with me, guys. Don't want you switching off too early. Okay, can you see my cursor moving about there, guys? Just type a yes. You can't see my cursor. Okay, that's interesting. Oh well, I will not point with my cursor anymore because we can't see it. Okay, that's obviously not what we want to think about. Okay, so imagine we've got the myosin heads, and they're basically like a golf club. Yeah, imagine you've got two golf clubs kind of twisted together. Can you see my cursor now? Oh, fantastic. Fantastic. So you can see there. Right, good. So here's the myosin fiber, okay, and it's got these two golf clubs, okay, and they're sort of wound together. Yeah, twisted together, okay, and they're going to be important for what happens in a minute. And hopefully we come up with, I've got a slide that shows you that in a minute. Okay, so we've got the thin filaments, and they're made of actin, and we've also got the troponin and tropomyosin, but it's actin and myosin that we're majorly concerned about with regard to the sliding filament theory. And then we've got the myosin binding site, and that's the myosin binding site, that's where the golf club is going to attach, and that's basically covered by the tropomyosin when the muscle is relaxed. Okay, so when the muscle is relaxed, we can see that this is elongated here, Z disc to Z disc. We've got our M line here, we've got our thick myosin filaments and our thin actin filaments sliding over each other. Okay, so we're going to talk about the muscle contraction and what you need to know with regard to muscle contraction in terms of the strength of a muscle contracting. So we've got the myosin cross bridges, and they're going to pull on the thin filaments, kind of like this kind of ratchet mechanism. And then the thin filaments are going to slide inwards, and this is the sliding filament theory. So effectively, imagine my thumbs are the Z discs, and the Z discs are going to come closer together. As the sarcomeres shorten, the muscle fibers shorten, and the muscle contracts. Okay. Just remember that the thin and thick filaments, so the myosin and the actin filaments, they don't change in length. Okay, we used to think that they concertinaed together. That's now not true. Okay, and they slide over each other. Okay, I think I've covered that to death. So that is the sliding filament theory. Okay, so we're going to go on to the sarcolemma, the T-tubules, and the sarcoplasm. So skeletal muscle consists of fibers that are covered in sarcolemma. And these fibers contain T-tubules and sarcoplasm. So T-tubules are like these tiny little inlets, and they basically going to spread the action potential, which is the electrical signal, to all parts of the muscle fiber. Because remember, muscle is conductive, muscle tissue is conductive. So sarcoplasm in the muscle cell is like cytoplasm, and it contains a large amount of glycogen, which will be used for almost like sugar for energy production, and then the myoglobin is for oxygen storage. Okay, so we've got basically stuff, we've got sugar and we've got oxygen, all the nutrients that the muscle needs stored within the muscle itself. So this is what a T-tubule or a transverse tubule looks like. Okay, and there's small invaginations of the sarcolemma into the center of the cell, and they're also filled with this extracellular fluid, and that's really designed to carry that action potential, that electrical signal, down into the cell. And then we've also got mitochondria. And mitochondria lie in rows throughout the cell, okay? And they're going to be using ATP during the muscle contraction. So the mitochondria are like the batteries of the cell, okay? But they also, we now think the mitochondria also have a brain, just like the nucleus, okay? So mitochondria is what we call like the powerhouse of the cell or the battery of the cell, okay? So moving on. Then we've got the sarcoplasmic reticulum. And the sarcoplasmic reticulum is basically where the calcium is stored. So we've got the storage of calcium, and when calcium is released into the cell, that's going to trigger the muscle contraction. Yeah, so we've got this chemical energy of calcium released becoming a mechanical energy. Remember, energy is just transferred from one form to the other. It's not destroyed, as it were. Okay, so this is now where things get a little bit confusing, because there's an order of stuff to know. Okay, now this won't be in the exam. It's background stuff, but it might be in the CAA, won't be in the Viva. Okay, because this is theoretical knowledge. So we're going to start with this nerve impulse, and this nerve impulse is going to reach the axon terminal, and the synaptic vesicles are released. Okay, that's acetylcholine sort of released into the cell. And this acetylcholine, which is a neurotransmitter, is going to diffuse into the receptors of the sarcolemma. Okay, and then sodium channels open, and sodium is going to rush into the cell. And the action potential spreads all over the muscle tissue and down these T tubules, these transverse tubules. Then from there, sarcoplasmic reticulum is going to release its calcium into the muscle cell or the sarcoplasm. And then this calcium is going to bind to the troponin, okay? And it causes the troponin tropomyosin complex to move and reveal its binding site, okay? And then that will allow the myosin head to bind to the actin, and then that's the contraction beginning. Now, I'm well aware that's just probably blown your mind, having to think about that, okay? And I completely understand that that's a lot of information. So I'm going to give you a little picture. Okay, unfortunately, that is meant to be an animation that's not working because I can't get stuff to play in Collaborate. So we go to the next one, okay, which is pictorial. So here we've got this electrical signal, okay, that's come down here, and this is stage 1. This is the action potential being generated. So action potential is going to release this acetylcholine into the muscle, okay? And then that's going to go down the T tubules, okay? And then we're on to the second part, okay, which is calcium being released from the sarcoplasmic reticulum. So as this calcium is released, okay, this is important because that's where the muscle contraction begins. And then the calcium ion is going to bind to the troponin tropomyosin complex, and that's going to change shape and effectively remove the block, allowing the actin to show its binding site, and the myosin head can then bind. So once the myosin head can bind to the actin, you can see that there, we've got this contraction, and we've got myosin cross bridges that kind of do that. Okay, I've got some great videos on Blackboard for a bit more of a pictorial representation about that. And what they're going to do is they're going to pull these muscle fibers, okay, into the center of the muscle, and then we've got this release of the energy, which is adenosine triphosphate, ATP, and this hydrolysis that's powering this sort of cycling muscle contraction. Eventually, when we want to go back to a relaxed state, the body will start removing that calcium via a system called active transport back into the sarcoplasmic reticulum. After that, all that electrical energy has stopped, that action potential has stopped. Okay, the troponin is then going to block its binding site, okay, and the muscle's going to relax. So that's quite a lot of information. Okay, that is a lot of information. So in order to understand that, what I'd like you to do is just go and watch. There's a 2-minute video on Blackboard. Okay, 2-minute video on Blackboard. Okay, and it is in week 11. So let me find it for you. So week 11, and I'm going to put that in the chat in a second when I can just find it. My computer's a bit slow. Here we are. So it's under muscle physiology, week 11. And then it will be muscle contraction, physical theatre. And it's 1 minute 20 long. Okay, so I'll put that link in the chat now. For you to just go and have a look at that video. So you can click on that. And what we're going to do is we'll meet back in the room in about 3 minutes time, guys. So don't go anywhere. Okay, we're just going to meet back in the room in 3 minutes. Please have a watch of that video because it makes everything a lot clearer. Okay, back in the room in 3 minutes, guys. I'm glad you've enjoyed the video. So we just let me know, give me a thumbs up in the chat when you have finished watching the video. Fantastic. Right, so what that's representing is effectively an action potential. What we tried to do is we tried to get you to remember this stuff because it's actually really complicated to remember stuff. So remember, like, an action potential is like someone organizing a party. Okay, so basically Will, who wants to organize a party in that video, he starts that action potential. That's like him putting an invitation out to everyone, maybe on Microsoft Teams, going, I'm about to have a party. I'd all like you to come to the party. So what happens is that action potential gets traveled down, so that message gets sent out to everyone in the group, so that everyone now knows what's happening. Then we get a small group of people, okay, who will be the party organizers, and that's like the Sarcoplasmic Reticulum, okay, and they're going to release their kind of enthusiasm and energy to the rest of the year about how amazing this party is going to be. That's like the calcium being released into the cell. Okay, so as we're doing that, so when the cell excites, okay, imagine for the guys, the guys are all being poured a nice pint of beer, and the ladies all being given a nice glass of Prosecco, or whatever they want to do. And when they're given the glass of Prosecco, effectively, imagine the men are the myosin, okay, and the ladies are the actin. And what happens is when the calcium goes into the cell, the lady's going to expose their binding site. And again, they revealed that elbow. And effectively, that's the tropomyosin, the troponin tropomyosin complex changing shape, allowing the myosin head to bind to the actin. And then we've got this power stroke as the muscle contracts. So the muscle will keep contracting, as long as there's enough calcium in the cell. Okay, once the action potential stops, okay, the binding site will be covered and the muscle goes back to its normal length. Is that easier to remember? Just give me a yes or no in the chat if that's easier to remember. Yeah, Rosie says, Rebecca says, yeah. Okay, so we try our best to make that interesting for you, and it's hard work. Obviously, with social distancing, we're unable to do that, but I thought it was still really relevant to show you because that's quite a nice way of remembering things. But believe it or not, okay, that took a whole about 45 minutes to an hour to get them to practice that and to do that. And once you've done that a couple of times, it will stay in your head. Very good, Harry. Harry, you're very funny. That's very good. Okay, I shall remember that. Okay, so now we're going to be talking about how does all this sort of muscle contraction, so what? Why do we need to know about that in exercise prescription? Well, obviously you need to know about human anatomy and physiology. That's what we do. That's what we understand. So what we need to do is we need to think about the acronym ICE, which is our isometric, our concentric, and our eccentric muscle contractions, and how they relate to the things that we need to do. So sometimes it's going to be easier starting with an isometric contraction. And we start with an isometric contraction normally in mid-range. Okay? And I'll tell you why we want to start in mid-range when we're doing like muscle strength testing, isometric muscle strength testing. So isometric strength training is a great way to carry stuff, like for the bicep. If you want to carry stuff, the bicep is in mid-range and you're holding something out in front of you. Okay, so the bicep stays at the same length and you actually hold something out in front of you. That's very functional for the biceps. Bicep curl, less functional, but actually holding stuff in front of you is highly functional. Then we're going to create this concentric force. So if the force generated by the muscle is greater than the external force, we can get a concentric muscle contraction. This is like me doing a bicep curl, okay? As I go against that, as providing I've got muscle strength to go against that resistance. Whereas if I'm lowering against the resistance, that's going to be eccentric. I'm lengthening under load. And if we want to strength train someone, okay, which muscle contraction would give us the best contraction for strength training? Concentric, isometric, or eccentric? Type it in the chat, guys. What do you think? Rosie thinks eccentric. What does anyone else think? Liam thinks concentric. Okay, so you want me to repeat the question. So if we are strength training, which muscle contraction is going to create the most muscle damage? And then because it creates the most muscle damage, when the muscle heals, it will repair stronger. Oh, there's a split between concentric and eccentric. So I'm going to rephrase the question. What muscle contraction will allow us to lift the greatest weight? Concentric, isometric, or eccentric? Yeah, it's going to be it's going to be eccentric, actually. It's going to be eccentric because if we're lengthening under load, okay, we're effectively just resisting gravity and the force. So eccentrically, okay, as the muscle lengthens, we're going to create more muscle damage. Okay, you might know this in the gym as negatives. Okay, give me a thumbs up in the chat if you've ever done negatives in the gym. Okay, where imagine you're bench pressing and you're letting the bar come towards you, and you're getting your hand pushing up and you're slowly lowering down. Okay, so what I want you to do is experience this in the form of a press up. So what I want you to do is lie on your tummy. Lie on your tummy. Everyone lie on your tummy. Okay, I'm going to do a visual. So you're lying on your tummy, and then what I want you to do, get everything tight, 5 pillars of stability, C-spine, scapula, midline stability switched on, tension in your glutes, pushing down through your big toe, and you push up. So as you push up, that is a concentric action, okay, of the pectoralis major. And then what you want to do is you want to lower down slow as you can, and that is the eccentric movement. Okay, we're lengthening under load. In this case, the load is the body. And then if you come up halfway and just hold it, so your arms are slightly bent and you should be shaking. Okay, that's the isometric contraction. Woo! Okay, stop there. Come back, have a seat. Give me a thumbs up in the chat. Let me know you're back in the room. Thumbs up in the chat when you're back. Hopefully you're not too exhausted from that. Okay, so we now understand the difference between concentric, isometric, and eccentric. So which was harder? What, pushing up, lowering down, or holding at mid-range? Which was harder? Yeah, should be the lowering. Rosie, okay, Rosie, I think you need to do more strength work. That's all I'm going to say on that one. Okay, it should be the lowering. The eccentric lowering is what we're after. Okay, no, you're just not strong enough. You're just not strong enough. Okay, so now we want to understand about length tension. So we're going to stay with the biceps because it's easier to understand. Okay, so when we are, okay, when we've got the muscle at outer range, so we're looking at the bicep. When I'm in this position with my elbow just slightly flexed, this is an outer range position. So if I'm resisting myself here, this is outer range biceps. Okay, and I'm particularly weak in this position because I'm too stretched, sorry. So there's less overlap of the cross bridges. Okay, so outer range bicep, I'm not very strong. You could also do this with the hamstrings. Yeah, okay. Now, if I go all the way in, okay, and resist myself pulling in, again, I'm not very strong. This is inner range biceps. Okay, and the reason being this is because I'm too contracted. So there's too much overlap. So the Z discs are now crumpling. Remember the Z discs? Yeah, but the Z discs are now crumpling on in each other. Okay, and they're blocking the process of me. I can't contract anymore because I've got all this crumpling here. Okay, so in here, okay, I'm quite weak at inner range and outer range. But if you test me at mid-range, that's when I'm strongest. Okay, mid-range means I've got the optimal cross-bridge formation. So I've got that optimal actin-myosin overlap. So that's why we tend to test in mid-range. Okay, so give you an example of this. Okay, it is the passive insufficiency. Okay, so what I want you to do is make a fist, squeeze your fist, and relax. Okay, and you should be able to make a strong fist. Even Rosie, you can make a strong fist. Okay, so what I want you to do now is make a fist and now flex your wrist, and you'll find that you can't make a very strong wrist there, can you? Okay, because of that passive insufficiency. Okay, just type in what do we mean by passive insufficiency in the chat box there, guys. What do we mean by passive insufficiency? You've done this to death in AMP1, I'm sure. Give us a nice definition of passive insufficiency. Yeah, you can't stretch if the joint goes over two muscles. Okay, so in the lower limb, give me an example of a muscle that will suffer normally from passive insufficiency. Rec fem, yes, rectus femoris. Okay, that's the classic one as well. And gastroc. Okay, we also, so gastroc rather than gastro. Gastro might refer to a pub, a gastro pub. That's a special dining pub. Okay, so gastroc is the abbreviation. Just need a C on the end of that. Okay, so that is really all we need to understand with regard to muscle physiology. It's important that we understand about inner range, outer range, and mid-range when we're prescribing exercise, because we need, so for example, what is the most important range when we're talking about rehabilitation of the quadriceps, for example? Okay, mid-range, inner range, or outer range? What is the most important thing if we want to get walking as quickly as we can? What range is most important? So we've got mid-range, we've got inner range or outer range for the quadriceps. What if we want to get walking? What is the most important range for us to work in terms of rehabilitation? Not quite, James. A good attempt. Not quite, Lauren. It's okay. No problem, Megan. No problem. Okay, so it's inner range quads. Okay, so inner range quads are going to help you stand, aren't they? So if somebody has got weak inner range quads, they can't stand up, which means that they can't walk. So those inner range quads, do you remember when we got, okay, a little ball and we're just doing the last 30 degrees of knee extension? So what I want you to do, you sat down, yeah? Okay, so straighten your leg out in front of you, bend it to about 30 degrees, and then just put your hands on your quadricep and up and down. So that is inner range quads. The quadriceps are working on their inner range, and that's important for walking. Okay, so when we're doing our rehabilitation, we would normally start with inner range quads because that's going to be the most functional for standing. Just give me a thumbs up in the chat if that makes sense to you. Thumbs up in the chat if that makes sense. Yeah, Joe's all over this. James, yeah, is happy. Liam's happy. Elliot's happy. Good, good, good. Okay, so what we need to do now is you're going to have a look at muscle contraction in some of the directed study. I've got some fabulous videos on that. As long as you understand what's happening in the physiology, we can go places with that, because that's that deep underlying process that underpins what we do, that we understand muscle physiology. Okay, so the only way that we can do that is obviously to test you in Kahoot. Okay, so just give me a second and I will load up Kahoot for you, and we do the muscle physiology Kahoot. So let me give you the code. Give me 2 seconds and I shall find it for you. Okay, last Kahoot of the year. All to play for, guys. All to play for. Where is it? Okay. Right, I think it is this one. So let's go. Right. Okay, so the number is 709671. 709671. Just type that in the chat. 709671. Hopefully you can get that in the chat, guys. Right, let's have a look. Yeah, we're going great guns. Okay, let me just share my screen with you. Okay, hopefully you can all see that. Yes, we can. Fantastic. Okay, let's get straight into Kahoot. Okay, we've got 16 in the room. $18 in the room. Come on, guys, it's the last Kahoot session. Make sure it's a good one. Fantastic. Give you a couple more. We've got 25 in. I want 5 more people, guys. 4 more people. Nearly there. Come on. Keep going. Stay with it, guys. Nearly there. I want another 3 people. I want 30 people in the room minimum. Come on. Let's keep going. Another 3 people, guys. Thank you, Holly. Another 2 people. And we can go. Another 2 of you. Sam's in. 1 more. We need 1 more person. And then we can begin the quiz. The Kahoot champion, Shana's in. Okay, oh, someone dropped out. What's going on? 2 people dropped out. What's happening? Come back. Come back in. There you go. That's better. Okay, we shall begin, guys. We shall begin. Here we go. Last Kahoot of the year. Oh, who's dropped out again? What's going on? What's going on? Come back in the room. We need 1 more. Come on. Who's coming back in? There we go. Here we go. First question. Which of the following is not a property of muscle tissue? It's obviously inevitability, isn't it? Because muscles are excitable, they're contractile, okay, and they're conductive. Well done, guys. Who's on top, though? It's Lauren, followed by Elliot, then Liam, then Becky, then Ellie Rose. Okay, next question. Question number 2. Which of the following is not a protein within muscle? Okay, excitable proteins. Okay, who got that? Lauren's still on top, then Ellie Rose moves up to second. Will, Zara, and James enter the leaderboard. Question 3. Which of the following is not part of the sliding filament mechanism of contraction? Okay, it is, of course, ozone. There is no ozone in muscles. Okay, Lauren is still on top with Ellie and Will hot on her heels. James is still in 4th, but Zara moves up to the leaderboard. Well done. Question 4. Which of the following ions are not involved with muscle contraction? What do we think? And it's, of course, strontium, which is radioactive. We don't want that in the muscles. That would be bad, bad, bad. Oh, look at that. No change at the top. No change at the top. Last question. No, we got two more. During muscle contraction, which one of the following is true? Okay, remember thick and thin filaments, the actin and myosin, they don't change length, they just slide over each other. Got to read the question, guys. That's caught a few of you out. Okay, Ellie Rose goes top. Okay, then James, then Zara, then Shauna, then Wendy. Okay, last question. Which of the following is not a type of muscle contraction? Okay, it's of course scalelectrics. Okay, if you don't know what that is, you're going to have to look that up on a search engine of your choice. Okay, so who is third place? Who's in third? It's Zara. Second place, it's James. James D. And well, winner is Ellie Rose. You are the winner for 2020. No one can touch you. Wendy and Shauna in fourth and fifth place, respectively. Big round of applause for Ellie Rose. Absolutely fantastic stuff. Very well done indeed. Okay, that is absolutely superb. Okay, so well done there. Right, okay. So what we're going to do is we're going to have a quick 10-minute break. So get yourself a cup of something nice. Have a little stretch, mobilize, maybe do a few squats, lunges, deadlifts. Okay, maybe some kettlebell swings. That's what I'm going to do. And we're back in the room in 10 minutes. Okay, so don't log off. Don't go anywhere. Just come back in 10 minutes time. Give me a thumbs up in the chat if you're happy so far, guys. Happy so far? Brilliant. Okay, so we're going to have a 10-minute break, guys. 10-minute break, and we'll see you back in the room for some revision. Thanks very much.